This video is going to go over the directions for the pGLO Bacterial Transformation Lab you will be doing tomorrow. This is a picture of the pGLO plasmid. Remember, a plasmid is a circular piece of DNA found in prokaryotic cells. We're going to use a plasmid to insert into bacteria, and that pGLO plasmid has to have an origin. It's the beginning point so that DNA polymerase can attach to it and can replicate that plasmid so that the bacteria cells can make more bacteria cells through the process of mitosis, cloning. On that plasmid, there's also an ampicillin resistance gene. We're going to use ampicillin on the plate so that it's going to kill off any bacteria that's not transformed. The bacteria that actually gets transformed with this plasmid will end up growing on the plates because of this resistance gene. So we're using it to select for that transformed bacteria. We're using it to our advantage. We also have the arabinose, which is a sugar, just like the lactose that you saw in the video earlier. Arabinose is a sugar, so that operon, when the sugar is in the environment, the operon is going to get turned on. Instead of just breaking down the sugar, though, we've tricked it and inserted something called the GFP, or the green fluorescent protein gene. This is coming from jellyfish that live in the bottom of the ocean that glow. We're going to be able to get bacteria to glow under UV light. Here are a couple things that you need to know. LB, when you see the letters LB, that stands for Lurry broth. It's just bacteria food. It's nutrients that the bacteria need, and it's going to be found on every one of the 4 plates that you are going to be getting tomorrow. When you see AMP, that is ampicillin, that means that there's going to be ampicillin on that plate. So there are 3 of them that have ampicillin, which is going to be the antibiotics. Plate A does not have the ampicillin. If you see a plus next to something, it's going to mean that we have introduced the plasmid to that plate, to the bacteria that's going to grow on that plate. If it has a negative, it means there is no plasmid that we're looking for the bacteria to transform and to take up and transform with. When you see the word arabinose, or the letters ARA, arabinose is the sugar. That's the sugar that's going to turn on the operon, right? It's going to bind to the repressor. It's going to make that repressor fall off. Now transcription of the arabinose gene can happen. But instead of doing the arabinose gene, we've replaced it with the GFP, that green fluorescent protein. So we're kind of tricking it to then make it glow in the dark. But arabinose sugar must be present in order for that to happen. There are 3 different results that could happen in this lab. Colonies, like this one here, are just going to be little dots on the bacteria plate. They can either glow under the UV light, or maybe they're going to grow, but they don't glow under the UV light. Another thing that can happen is a whole overgrowth of bacteria. We call it a lawn of growth. A whole bunch of growth all over the plate. Another thing that can happen is absolutely no growth on the plate. Bacteria, there's absolutely no growth. A tool that you'll be using is called a pipette, and it's not like the ones we've been using. These are disposable, so you open it kind of like a Band-Aid, like the picture on the left. You open it like a Band-Aid, and you'll have a sterile pipette. And we need these sterile pipettes. We're going to make sure that we don't have any cross-contamination with anything. So we do need to use different pipettes every time we do a different step. If we look here on the right, there are different measurement markings. Right here would be the 100 microliters. This is 250 microliters, or 0.25 milliliters. If you go all the way to the top here, that's 1 milliliter. So if you're ever stuck, go back to this and see where you need to squeeze that pipette, right? You're going to put your fingers and thumbs right here to squeeze that pipette so that the volume of liquid goes up to wherever your desired amount is. This is a picture of the basket that you're going to have at your lab table. Here are all of those squeezable pipettes. Okay, those are the sterile pipettes that we're going to use. You have a permanent marker, you have tape that we're going to use to do some labeling. You also have these inoculation loops, and for cross-contamination, we're going to be using different inoculation loops to plate the bacteria. Another thing that you're going to have is a bowl. This is actually going to be filled with ice. There are a couple things that we need to keep on ice. You're going to have a tube that says TS. This is going to stand for Transformation Solution. The LB stands for the Lurry broth, which is that bacteria food. You're going to have a pink tube and a green tube, and those colors are going to be important here in a little bit. Now, here are the steps for the transformation lab. You need to pull up your lab and scroll down to the bottom where you start to see this and split screen this video with those directions that you have in your lab. Anything I have written down in this video, you should be writing down on your lab. So when we get to this lab tomorrow, you know exactly what to do. The bacteria that we are using is E. coli. And I had mentioned that there are two colors to these tubes. You're going to have a green tube and a pink tube. The green tube is going to have no plasmid introduced to it. We're not going to try and transform that bacteria with the plasmid at all. The pink tube is going to be a tube that we introduce the plasmid to, so it's going to be positive. So think of P for pink, P for positive, P for plasmid. The other thing we are going to have on here is the Lurry broth, the nutrient broth. It's going to be nutrients for the bacteria to grow. So the first thing you need to do is label the pink plasmid with a positive at the top and a negative sign on the green one, so you know which one is which. You're then going to open up the tubes, and with a sterile pipette, that transfer pipette, you're going to pipette 250 microliters of that transformation solution. It's a CaCl2 solution. For chemistry, I think it's calcium chloride. And it's just going to make sure that we are able to transform this bacteria. It gets them ready to open up their cell membranes to allow that plasmid to come in. So that transformation solution, which is labeled TS, 250 microliters are going to go into your pink plasmid, and 250 microliters are going to go into that the green tube. I think I said plasmid for the pink. It's a pink tube and the green tube. Those tubes then need to be placed on ice as we're working on the next steps. You are going to have a plate that has a ton of bacteria on it. Using a sterile loop, the yellow inoculation loops, you're going to pick up one single colony of bacteria, so a dot that you see on there. You don't need to swipe the whole thing. That's way too much bacteria. Remember, bacteria are going to grow very rapidly over the course of a few hours. We're going to have lots of bacteria, so we only need a little bit. So pick up one colony with that inoculation loop. You're then going to put that inoculation loop with the colony of bacteria into the pink tube that has that transformation solution. And you're going to twist your fingers and thumb in it so you get all of that bacteria into the liquid. You are also going to do this with a new inoculation loop to the green tube. And again, twirling your fingers and thumb so that you get all of that bacteria into the transformation solution. Now, be careful when you scrape the bacteria off of the agar plate. Be careful not to break the agar. It's like a jello solution, kind of like that gel electrophoresis, the gels that we've been doing in the past. So it's really fragile, so just scrape it ever so gently. You do not want to dig. You're going to place those tubes back on the ice until we're ready for the next step. When you're ready, you're going to bring me just the pink tube up to the teacher lab table. I will have the plasmid DNA, and I'm going to take an inoculation loop, and I'm going to put that plasmid with the inoculation loop and the plasmid into your pink tube. So I'm going to twirl around, and I'm going to put the plasmids that we want the bacteria to take up into your pink tube. Do not bring me your green one. I don't need it. Those are the ones that don't get exposed to the plasmid. We then need to incubate all of those tubes on ice for 10 minutes. Make sure that you've pushed all of these tubes so that the liquid is in the ice for those 10 minutes. While the tubes are sitting on ice, there's a few things that you can do. Make sure that everything is labeled. All of your plates are labeled with a positive pGLO, with the LB and AMP for the nutrient broth and the ampicillin. You should also have one that's going to be positive pGLO with the LB, AMP, and AURA. Those are coming from your pink tube when we're ready to swipe it on there. 2 of the plates are then going to be with your green bacteria, the negative pGLO, the LB and the AMP, and the negative pGLO LB. So you actually have 2 plates that are LB AMP, but one's for the positive plasmid bacteria and one's for the negative. Also put your lab table number on it. And all of this should be pretty small because you are going to want to be able to see through the plastic when we look at these the next day. Also, while you're waiting for those 10 minutes, if you need time to work on the pre-lab questions, if you need to be working on your paper plasmid lab, you have some time to do that. This next step involves a heat shock. We want to make sure that the bacteria kind of opens up the cell membrane and with the help with the transformation solution and this heat shock. So it's going from ice to a water bath back to ice really quickly. You want to bring your whole entire ice bucket with you back to the water bath so that you can transfer those tubes into the 42 degrees Celsius water bath for 50 seconds. Use your iPad timer. After those 50 seconds, right away back onto your ice. That's why you want it next to you. Then you can go back to your lab table, another 2-minute timer. Those tubes are going to stay on the ice. Make sure that the tubes are actually touching the ice. After those 2 minutes, you're going to take a new pipette for each one and squeeze in 250 microliters of the LB nutrient broth into the pink tube, and then a new tube, 250 microliters, into the green tube. We then need to incubate at room temperature for another 10 minutes. During those 10 minutes, again, you could be working on your paper plasmid lab, or if you still need to finish up the pre-lab questions for this lab. After those 10 minutes, you're going to take a new pipette for each time, and you're going to put 100 microliters, so a little amount, onto each of the plates. If it's a positive, that's coming from your pink tube. So 1 of these, 2 of these are pink. New pipette each time. We don't want any cross-contamination. Then you're going to open up your green tube, and you're going to pour 100 microliters into each one of those 2 as well from the green tube. Make sure you're doing the right ones. Look at your labels and make sure it's all correct. Once that's finished, you are going to cover them and flip them upside down so that when I put them into the incubator, if there's any condensation, that it's not going to drip into your agar plate, just in case there was any bacteria on the plastic lid. So you're going to stack them upside down and tape them together and put your lab table number on the tape. I will then take care of them, put them into an incubator. We'll incubate them for about 24 hours and hopefully we have results for tomorrow. Hopefully these steps were pretty clear. If you have any questions, please don't hesitate to ask. This lab is going to go bell to bell when we do this, so that there wasn't any time for me to explain this lab during that. But if you have any questions even before class or real quick beforehand, please ask for clarity. There's no shame in asking for help with that. And let me know anything I can do. Thank you for watching.